Good evening, everyone. My name is Anthony Lamantia. I am the director of the Center for Neurobiology Research at the Fralin Biomedical Research Institute. And tonight it's my pleasure to welcome you to the first Maury Strauss Distinguished Public Lecture of the 2024-2025 academic year. This is our first lecture following the passing of Mr. Strauss this past summer. We're very honored that Mr. Strauss's daughter, Leslie, is here with us tonight. And of course, we are extremely grateful to Mr. Strauss's generosity and the generosity of the Strauss family over the years, supporting this very important activity for the Fralin Biomedical Research Institute. I want to remind everybody that the next Distinguished Public Lecture is October 12th, a Thursday. Dr. Vera Gorbanova, the Doris Johns Cherry Professor of Biology and co-director of the Rochester Aging Research Center at the University of Rochester, will be here to tell us about mechanisms of longevity from mice to whales, and I'm hoping she fits in humans somewhere in between the two. All right, tonight, I have the distinct honor and pleasure of not only introducing, but in a way reintroducing you to our distinguished public lecturer, Dr. Michael Friedlander. Many of you know Dr. Friedlander as the director of the Fralin Biomedical Research Institute, as an indefatigable advocate and exemplary citizen of the Roanoke community, a tireless advocate for biomedical research, its health benefits, as well as its educational and economic opportunities for Roanoke and the region. But what many of us don't know as thoroughly perhaps due to Mike's own modesty, is the impactful trailblazing research that he has pursued in his laboratory over the course of his career. And my job this evening is to introduce you to Dr. Friedlander in that context. Now, there's a personal note here because I first met Dr. Friedlander in 1982 at the Society for Neuroscience meeting when I was a lowly first-year graduate student presenting my undergraduate research in a session where Dr. Friedlander, an acknowledged star in the field, and his trainees were also speaking. I was introduced to Mike Friedlander by one of my colleagues. I'm not sure he remembers me because, there was a throng, but I certainly remember him, and so it is really remarkable how time leads you to where you land. Dr. Friedlander's stature as a leader in neuroscience of his generation reflects his elegant, creative, and pioneering work published in the most influential journals in the field, including Science, Nature, Cell, Proceedings of the National Academy, Neuron, and the Journal of Neuroscience, that led to true mechanistic understanding of how the brain works to generate and store experience, what the philosophers call the hard problem. He was instrumental in laying the foundations, both conceptual and technical, of how neuroscientists look at single neurons to understand how they interpret what's going on in the world and how they contribute to larger networks to build an integrated representation of what we see and what we think. He also helped set the stage for ongoing efforts to understand how brains retain the capacity over time to change in response to new information processing demands, giving us the flexibility that we all value. Dr. Friedlander and the many trainees who began their own successful careers under his guidance focused on how key neuron classes in the brain, particularly in the visual system of the brain, use their cell biological and molecular capacity to receive and generate electrical signals to represent information about vision, what we see in the world around us. He also helped define how neurons change to accommodate new experience via altering those same cellular, electrical, molecular, and synaptic properties. His work has also influenced how we think about the way key chemicals in the brain, including nitric oxide and calcium, which he will tell us about tonight, act to modify a nerve cell's capacity to process information and work together to better guide perception and behavior. His talk tonight, Conducting the Neuro-Symphony in the Brain, space, time, and calcium, will provide an overview of how Dr. Friedlander has led us to think in new ways about these fundamental questions. Now remember, he did all of this while he was a professor at UAB, the chairman of the neurobiology department there, a professor at Baylor College of Medicine, the chairman of the Department of Neurobiology there, and then the founding director of the Fralin Biomedical Research, the inaugural vice president for health science at Virginia Tech, the inaugural dean for research at the Virginia Tech Carilion School of Medicine, and truly a tireless supporter of science, of the careers of all of us who are here, the students' progress here, and of the Roanoke community. He also had time to be Sandra's husband and father to 5 daughters and 9 grandchildren, so he never sleeps. So here he is, wide awake, Mike Friedlander.
Well, let me turn this on. Money came out of my pocket with it. That's a rare thing, as my wife knows. Hello, can you hear me? Great. Thank you, Anthony. Extremely kind words and not deserved, I'm sure. So it is a pleasure to be here. It's unusual for me to be on this side. Usually I'm doing the introductions, but it's an honor and a pleasure for me to tell you a little bit about the work that's gone on in my own lab over the years. And I have a little story I want to tell you, but I want to make sure at the beginning to tell you, as you all know when it comes to science, these stories are always about a lot of other people, not the person who's standing up here. So these are some of the key students and fellows that have worked with me in my lab over the years. I'm not going to have time to point out every one as we go, but I wanted to get them up there. I'm extremely proud of all of them, many of them now in their own very important positions. Laura is a professor at Tulane University, Prakash is a professor at the University of Minnesota, Reed is a professor here, as many of you know. Serena is a professor at NIH at the National Institute for Environmental Health Sciences. And interestingly, Rod is a pilot for Air New Zealand. So a lot of interesting different career trajectories for these folks. But nonetheless, they're the ones who did all the work, and that's what I'm going to tell you about today. Also, very important for me to acknowledge the people who supported this work, you. Not often enough, I think we don't acknowledge US taxpayers who support research in this country. That's all of you. I assume you've all paid taxes, I guess. And for the research in my lab, it's been the National Institutes of Health, particularly the National Eye Institute. Some of the work I'll talk about today was supported by the Department of Defense, the congressionally directed medical research programs, and I'll briefly mention some stuff supported by the National Science Foundation. So that's where the money comes from and the support to do all the work that we've been able to do in my lab. So I want to start with this little primer to talk about the different types of research that go on in the biomedical and health world. There's what we call basic research, translational research, and clinical research. I'm going to concentrate, I won't bother to read these to you, you can take a look at them. I'm going to concentrate on this end, basic research. That's primarily what my own lab does, but I want to make the case, and hopefully I'll convince you how fundamental that is and leads into these next stages of translational and clinical research. And you don't always have to have that clinical question in front of you at the moment to start to discover things that are important and fundamental that others can pick up on and carry forward and translate it and even carry it into the clinic. So I'll try to make that case for you as we go forward. So this is the outline of my presentation. It has, what's it, 5 different parts. First of all, just a little background about how I got into all this stuff, a little bit about serendipity, opportunity, mentorship, and yes, fish. And then a very brief primer on how synapses work. And then I'm going to talk about this section, timing of electrical activity in the brain, and how important it may be for information processing and in carrying it forward to clinical care. And then what I call space, where these things are in the brain, synapses, and how they distribute themselves and how they talk to each other. And then finally, this kind of crazy thing I say, is calcium really calcium? The nuances and roles of this multifaceted molecule when it comes to plasticity in the brain and function in the brain. So that's the goal of the talk. In terms of what's downstream, in terms of potential clinical applications, I just want to emphasize there are many neuropsychiatric disorders that are linked to dysregulation of synaptic plasticity. These are just a few: substance abuse, Alzheimer's disease, autism, depression, Fragile X syndrome, PTSD, and schizophrenia. These are all people that many of you would know who have suffered from these things, John Nash, Daryl Hannah, and so forth, President Reagan. And there are, things that can affect all of us or any one of us at any time. And these are things that we know are directly related to synaptic plasticity dysfunction. And also, when it comes to my third topic, calcium, we know a number of disorders: ischemic stroke, again, Alzheimer's, Parkinson's disease, multiple sclerosis, epilepsy, and leukodystrophies also can be related, at least downstream, to dysregulation of calcium in the brain. So these are things that, having a better understanding what's under the hood, what's going on, may be able to direct us towards trying to help in those areas. So let me just start with the first section, a little bit of background on how I get into this. So I was an undergraduate student at Florida State University. I was majoring in political science and economics, planning to go to law school. I took a biology course that this gentleman taught, Dexter Easton. I had some questions. I talked to him after a class. He said, "Why don't you come by my lab and I'll tell you more about it." Next thing I know, I was in his lab and I was doing studies on these critters called Lepisosteius osseus, the long-nosed garfish that has a long olfactory nerve. And I was studying how information is processed by smell in this fish. Next thing I knew, I was taking all these courses I needed to be able to work with all this equipment in physics and math and chemistry. Somebody said to me, "Well, it looks like you've done a pre-med curriculum, but you're going to law school." And I toyed with the idea, maybe I would go to medical school, but I knew that's not really what I wanted to do. But I got excited that somebody told me you can actually make a career out of doing research. I had no idea, none whatsoever. I didn't have any role models in that respect. But that's what I got into. And next thing I knew, Dr. Easton called this gentleman, Lad Prosser, who is somebody who literally wrote the book. This is one of his 12 editions of a book called Comparative Animal Physiology. He was at the University of Illinois. He invited me to join his lab as a graduate student in Champaign-Urbana, Illinois. And there I got involved with his work, and it also included fish and included something at the time that was pretty interesting. It was temperature changes and how it affected the brain. And at the time, and I'm dating myself here, this is going way back, when people were worried about climate change where everything was supposed to get really cold, my thesis project was on what happens if things get hot. Everybody thought I was crazy. The world wasn't going to get hot. It was all going to freeze. We were going to have nuclear winter, et cetera, et cetera. And so I started doing this research on biochemistry and physiology of synapses in the brain of animals that would be living in the environment when it warmed up. And that's how it all started. And we published these two papers. Thank God you can't see the years because you would know how old I am. But nonetheless, these two papers where we studied the chemistry of synapses in the brain at different temperatures and how that affected fish. And also we studied the neurophysiology, the electrical activity in the brain as a function of temperature. Now, I'm sure almost nobody's going to recognize these. This is a computer that I used in the lab at that time called a Digital Equipment Corporation Link 8 computer. It had little tapes that went back and forth and switches and lights that were flashing. It was really exciting. It took up half a room. It was hot as heck, and it didn't have 1,000th the power of the iPhone that I'm carrying in my pocket. That's what I used for my dissertation research. This is an old teletype. This is what we would use to print out the data coming out. It used to go, and the thing was jumping around on the floor and it was making dots all over the place. And these are the way we plotted the data. So what I'm showing you here is the time between electrical impulses in a neuron in the brain of a fish. Happens to be a goldfish, happens to be the cerebellum. And you can see the time intervals at these temperatures are pretty tight. But then notice here, at a very warm temperature, these fish had grown up at, and these are degrees centigrade, these fish had grown up at warm temperatures. Notice how the intervals are all distributed widely. What that means is the neurons aren't going bip, 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 bip. They're going bip, 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 bip. They're kind of going crazy. And I'm listening to this on a loudspeaker. It has stuck with me all those years. What the heck is that about? And what does it have to do with anything? And we looked at the chemistry of the synapses to try to understand it. I totally forgot about this stuff. I went a totally different direction, studying synaptic plasticity in the brain and mammals and so forth. And then a few years ago, this article comes out from this gentleman, George Summerow at Stanford University, who talks about these papers and calls them a classic and said they've changed the whole way we're looking at the field, et cetera. I mean, nobody had read those papers as far as I know, other than my mother for 30 years or 40 years. And decided with climate change coming as it was, and with warming, having some insights and understanding how increasing temperatures affected activity in the brain was going to be something important. So it got some more attention. Well, just to round out the fish world, and I promise I leave fish after this slide, When I was at the University of Illinois, Dr. Prosser had a lab at a place called the Woods Hole Marine Biological Lab in Massachusetts, and I would go out there in the summers with him to carry on our research. And one of the things we did was we would anesthetize sharks and put electrodes in their brains and record the electrical activity from neurons in the sharks' brains while they were free swimming. We developed a hydrophone and a transmitter system in Illinois, and we tuned it to a frequency called a carrier frequency in Illinois to broadcast. Little did we know, when we got to Woods Hole and put the sharks in the water, we had tuned to a frequency of a radio station in Massachusetts. So as the sharks were swimming around, supposedly transmitting the impulses from their brain, we were listening to actually country western music coming in over the speakers. So that experiment went nowhere, basically. But then to just come full circle and wrap up the fish story, several years ago, I got a call from some folks from the National Science Foundation, and they said, we're worried about global warming. We have an expedition planned to go to Antarctica where the waters are warming, and we need somebody who's an expert in studying what happens to fish's brains when the water warms up, and particularly what are called Antarctic ice fishes. And so a group of people from my lab, Ishkander Ismailov and a medical student, Jordan Sharping, who's now here at Carilion Clinic, I trained them up and they went down to Antarctica, worked with these very unusual fishes and studied what happens when their brains are warmed up as well. So this has kind of gone full circle for me, this whole thing about temperature affecting plasticity in the brain and activity and behavior of these animals. And as I said, it was just the side and what started me off. So now I want to just jump to what I've been doing the last number of years and focus on the main questions. But let me start with this little primer that I promised on synaptic plasticity. So obviously, this is a brain, this is a neuron. You always hear these numbers, how many tons of neurons there are in the brain, and then you hear about these numbers of synapses. So something like 10,000 synapses on an individual neuron. So when you think about computational capacity in a brain with literally billions of neurons, nerve cells, and then tens of thousands of synapses on each neuron, the question arises, if you're a computer scientist, is where does the computational capacity live? Is it the neuron itself, the cell that's the computer, if you will? Are those individual synapses, do they have a mind of their own, and are they computing things independently? And that's a question we've been really interested in that I'll address in a moment. And this is just a picture of a synapse, the front end or the presynaptic side, the back end or the postsynaptic side. These little circles here are synaptic vesicles that are filled with a chemical called a neurotransmitter. And the chemical we study primarily is one called glutamate, which is the primary neurotransmitter that's excitatory in the brain, including in the human brain. So here's the little primer. This is a drawing of a nerve cell or a neuron. This is the output, the axon. You see all the little pluses and minuses reflecting the electrical activity. You see how it changes as the nerve impulse races down the axon. And then eventually the nerve impulse, or what's called the action potential, shown here, the change in electrical activity reaches the end of the neuron where the synapse is. And then at that point, when the action potential gets here, here it is, the synapse releases the chemical neurotransmitter, and that bombards the postsynaptic neuron. These little blue things are called receptors, and the chemical binds to them. Once it binds, it creates the activity in the next neuron in the chain, and that's synaptic transmission, where an impulse gets converted into what's called a synaptic response, recorded electrically in this cell. Now, there's a variety of ways you can change the fidelity of this. And one of the ways you can do it is to kind of trick the cells, if you will, into having what we call correlated activity. And if you do that, the synaptic response will go up. We call this long-term potentiation, or LTP. The synaptic response gets bigger. Or you can do other things and make the synaptic response go down. We call that long-term depression. This is not the psychiatric term depression. This is depression of a synapse that gets weaker. It's not as effective at transmitting information. It gets smaller, basically. And so when you think about what might happen to cause this, there are a variety of possibilities. So here we have the picture of the normal synapse, the cartoon. Now we've done the trick to strengthen the synapse, potentiate it, create LTP. So in the cartoon, this shows several things. One, it shows on the postsynaptic cell, there are more receptors, more sites to capture the chemical signal. We know that happens. Two, it shows more of the chemical neurotransmitter, the little red dots coming out. We know that happens. So both a pre and a postsynaptic effect, changing the synaptic strength. So if you think about it, I think we have a little movie here that's supposed to, what am I supposed to do, Brad? Just click?
1,000 ping pong balls just to successfully hit the inside of the glass. And
Look
you
at all
will
those
never guess what
neurotransmitter
happens.
molecules
Now
coming
look,
down
the
there.
moment they drop the
Look at
balls,
that
they are
receptor,
certain
the
that at
glass.
least
Is
one
he
of the
gonna catch
balls
it?
will drop into the glass. But the balls seem to have their own life, jumping everywhere except in the glass. But the guys still have hope to successfully complete the challenge.
So
they
the way
watch
to
with
capture
intense
all those
eyes,
molecules
hoping
is
to
have a
make the
bunch more
shot. Their
glasses
hope is
out there,
fading away until a
receptors to
white
receive
ball
those.
moves towards the
Even
glass
if you're
and misses
releasing
it
more
inches away.
neurotransmitter, it's diffusing and bouncing around, basically. And so that's the idea. So let's have a look at some experimental approaches to understanding how this works in 3 domains. Domain number 1, what about the timing of the electrical signaling? Is that information useful, and how does it contribute to synaptic plasticity? So now instead of a fish, we're dealing with a mammal. This is a mouse, but the work could be done with a rat or a guinea pig, but these experiments are done with mammals. These are pictures of action potentials. Notice how perfectly regular they are, spacing time-wise. That's not the way it works. That's what you see in textbooks. But if you put a wire in a real living brain, including a human brain, and you listen to the electrical impulses, it doesn't go boop, 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 boop. It goes boop, 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 boop. It's noisy as heck, like this. These are the noisy patterns of action potentials that are firing in the brain. So we designed an experiment to see how that contributes to synaptic plasticity. So what you're looking at on the left here is an experimental protocol. We test the synapse at very low frequency, once every 10 seconds. Then we condition it over a period of time, stimulating at a certain frequency and pattern. And the patterns can be perfectly regular, or they can be slightly irregular. Maybe you can see little spaces in here, or they can be highly irregular. If we plot the time between the pulses, these are perfectly regular. They're all in one bin. Here you can see they're distributed, what's called a Gaussian function. And here it's a statistical, what we call a Poisson distribution, very irregular. So I'll just play those for you to listen to the stimulation. This is the perfectly regular one. I'll get this eventually. Oh, good. And here's the highly irregular one. So that's what you hear, and we do hear these things. We hook these electrodes up to computers and to audio monitors in the lab to listen. And a neurosurgeon does the same thing when they're implanting electrodes to treat neurological disease. They'll implant electrodes and listen over a loudspeaker to what we call the spiking pattern of activity. So in these experiments, we run these different protocols. How do I get beyond this? Oh, I'm beyond it. Okay, thanks. So here's an example of one such experiment. What we're plotting on the y-axis here is the size of the synaptic response. It's been normalized, so baseline is just 1.0. And then this is where we're stimulating. In this case, we're stimulating once a second, and this thing called the coefficient of variation is 1, which means it's the highly irregular pattern. And then we stop, and then we simply ask what's happened to the synapse. And what's happened in this case is the synaptic response got smaller from gray to orange, as you can see here, and it stays smaller. So this is an example of long-term depression. A period of time of an irregular stimulus in this neuron leads to depression. Well, what happens in these different parameters? So what you're looking at here is perfectly regular, a little bit irregular, and highly irregular at 1 Hertz stimulation. And here you're looking at perfectly regular, a little bit irregular, and highly irregular at 10 Hertz, 10 times per second stimulation. And you can see there is a difference here. So in this case, the synapses could detect a slight irregularity in the pattern. The frequency is the same, the number of pulses is the same. The experimental paradigm is identical, except a little noisier pattern. And what happens downstream is the synapses say, wow, boom, we're potentiated, that's LTP. So somehow those synapses can detect and read the degree of regularity. And the mechanism of that is an open question. And I just want to mention at this point, getting back to that earlier slide about going from basic science to translational to clinical, brain stimulation is being used in the clinic now in quite a lot of different ways. So what's called deep brain stimulation, or stimulating with electrodes in patients, is used to treat Parkinson's disease, but you can see the whole list of areas it's being tried in now. And in addition to deep brain stimulation, there are many other modalities, what's called transcranial magnetic stimulation, non-invasive things like focused ultrasound. So there's a whole bunch of different technologies out there now, and there's a whole bunch of disorders that physicians are using to try to treat these disorders by stimulating certain sites in the brain. And I'll suffice it to say, there's some very encouraging studies. There are some that are very positive in general, been clinically approved, such as in Parkinson's disease. There are others that are highly experimental. But this is a way to think about getting to modulating brain activity without dumping a drug on exogenously, but activating the brain's own circuitry to release its own chemicals. The thing about this is I found in talking to many neurosurgeons who are doing this is for the most part, they don't look at the timing or the pattern at all. All they do is stimulate at a frequency, like 10 hertz or 100 hertz. Brrr, everything equally spaced to get the effect. And so one of the things we're trying to do is to convince our clinical colleagues to begin to think about the timing and the pattern because of its effects downstream on the plasticity of the circuit after you stimulate it. And so this is supposed to be a eureka moment, I guess, like, gee, maybe this could be the greatest thing since sliced bread. My wife and I were watching a show on TV the other night about the history, I think, of McDonald's and fast food, and they had a story about bread slicers and sliced bread. So I really got enamored by bread slicers, honey. That's where this came from. So anyway, so all right. So the question is, can these help in the injured brain? So the next part of this experiment was, let's do our experiment on the timing and plasticity, but not in a normal brain, in an injured brain. So this is a laboratory experiment with an animal in the lab, a rat, that's had a very mild traumatic brain injury under anesthesia, then recovers. The animal looks perfectly fine, except for maybe a little bit of instability when you test them closely. And then we already knew that traumatic brain injuries, particularly mild ones, can often be devastating, but there can be very subtle effects as well. It's not like you're just looking at massive damage in the brain. There are subtle things happening in how neurons communicate with each other. So we decided to look at this in those animals as well. So what you're looking at here is the same picture I showed you before in a normal control animal, same stimulation parameters. You see the depression. Here we have what we call a sham. This is an animal that underwent the surgery but never actually had the injury, just to make sure the surgery itself didn't cause an effect. And then this is the animal with the brain injury, mild traumatic brain injury. Same parameters, and look how different the response was here. This case, the synapses strongly upregulated or potentiated, whereas in these cases, they depressed. So we already had a hint there was a difference in the injured versus the normal animals. And this is a very busy slide, but it summarizes a lot of the data. So once again, across the top here are the different parameters: perfectly regular, a little bit irregular, highly irregular control animal. This is the brain injured animal: perfectly regular, a little bit irregular, highly irregular. In this case, you can see the highly irregular pattern in the brain injured animal upregulates the synapses, makes them stronger. Why might this be of interest? Because downstream from a traumatic brain injury site, there's often a weakening of connections and communication in the brain. So if they can respond to these patterns by upregulating an otherwise downregulated pathway in the brain, it may have some clinical value. And these data are simply summarized here. And this just shows the same thing for the 10 hertz stimulation. Same exact thing: normals, brain injured animals, perfectly regular, slightly irregular, highly irregular. Once again, with the highly irregular pattern, the injured animals upregulate plasticity. So there's something about the injured brain that can detect these noisy patterns of activity, and something reads it, in the postsynaptic neuron can read that noisiness and turn it into a downstream molecular signaling pathway that changes the synapses and makes them stronger. Now, what effect that would have in terms of behavior and so forth remains to be determined. But we're very encouraged by these results. And these were just published a month or two ago. And I should mention Quentin Fisher, who's in the audience here somewhere, is the first author on this paper as well. And then the question rises, so why is that? What is it about an injured brain that takes a noisy pattern and turns it into upregulation versus downregulation? So one of the things we looked at was the calcium, which will transition to the next part of the story. These are measurements of the calcium in the neuron in the control animals, in the neurons, shams, and the brain injured animals. And you can see the resting calcium levels in the cell in the brain injured animals are much higher. Why is that important? Because we already know that calcium is an essential step to trigger plasticity and learning in the brain. Without calcium, it's not going to happen. We also know calcium can be deadly in the brain. Too much calcium in a neuron activates processes where the nerve cell literally eats itself up, digests itself. So calcium is deadly at certain levels, but essential at other levels. So neurons go out of their way to precisely balance and regulate the calcium inside the cell. And so these measurements in the brain-injured animals suggest us they may be sitting closer to a threshold to trigger upregulation or synaptic plasticity. We don't think that's the whole story, but at least it gives us a hint. And then I want to this is a little complicated, but I want to show it because it starts to get us to a mechanism. When you think back to all those ping pong balls and they were all being released, imagine they were neurotransmitter molecules. So we did a study in our lab. Zhangping Wu, a very talented postdoctoral fellow, was in the lab for a couple of years, did this work. And what he did, you're looking at a picture through a microscope here of a bunch of different neurons. And these little lines you see, these are the hollow glass tubes called micropipettes that we attach to or patch on the neurons. And so they're recording from multiple neurons at the same time. So what he's doing is looking at the effects of regular and noisy stimulation from one cell to one other cell. So notice it says A, B, C. This is neuron A, neuron B, neuron C. In this case, he's firing an action potential in A and recording the response in B and C. You can see it gives a response in B, but none in C. In this case, he fires an impulse in B, records from A and C. In this case, in C, and records from A and B, does it every which way. So we can actually evaluate at the individual cell level what's going on. Why is that important? Because when you analyze it that way versus stimulating the brain the way I've been showing you, where you stimulate lots of cells, you can actually infer mechanisms that underlie the process. In this very busy slide, I'm just going to call your attention to one part of it. So the blue versus the red are showing whatever it is we're measuring here in the preconditioning to the post-conditioning period. And I call your attention to this one area right here where it says FR. FR stands for failure rate. And failure means the failure of the presynaptic neuron to release neurotransmitter. Even though there's an impulse in the presynaptic neuron, an action potential, and the way the textbooks explain it, when that happens, it releases neurotransmitter. Not all the time. As a matter of fact, for many neurons, not even most of the time. They're fickle. The synapses are very fickle. And in this case, what's happened, because they've analyzed the single cell, is we've shown the failure rate went up. So this is what we call a presynaptic mechanism. The reason that's interesting is the plasticity is triggered in the postsynaptic neuron, but this is the presynaptic neuron that's changing. And synaptic transmission is supposed to go pre to post, not backwards, post to pre. But something has happened here, and Anthony alluded to it. I'm not going to show you the work, but we had done some work earlier on a molecule called nitric oxide that's made in the postsynaptic neuron, can go backwards and can change the release of neurotransmitter in the presynaptic neuron. And that's the same nitric oxide that is upregulated with Viagra and a number of other medications. What it's known for is dilating blood vessels, but we also know that it's in the brain. Years ago, we did an experiment that we published in a journal called Science. This was work done with Reed Montague, where we had part of the brain connected to the heart. And we stimulated the neurons in the brain to make and release nitric oxide that then diffused down to the blood vessels of the heart and dilated the blood vessels, showing that nitric oxide is indeed made in the brain. And others have gone on to show that nitric oxide signal can move backwards across the synapse and increase the release of the amount of neurotransmitter. So we think this is giving us at least an initial insight into process. So summarize that part, these different frequencies of stimulation. There is a difference in plasticity between the groups with the controls and the brain injured ones, particularly for the very irregular patterns. And in controls, there was also a difference across the regularity of stimulation. So we have some beginning insights into that. So now let me tell you the story about space and location of the synapses. So this is a neuron through a microscope with one of these little pipettes attached to it. And in this particular experiment, what we're going to do is activate different synapses onto the same neuron at different places and ask the question whether it's the synapses that behave independently to show this plasticity or whether the neuron controls the plasticity for all of its synapses. So how local and at what fine a scale is the control of plasticity? So again, I apologize, rather busy slide, but I'll hit the highlights. This is a picture of one of the neurons and the pipette on it has a fluorescent dye in it. This is a section through the brain where the neuron was. This shows the two different sites that were stimulating different inputs to the same cell. we go through a pretty detailed test to make sure we're activating different inputs of the same cell. And I should add, this is the dissertation research of Olivia Fitch as well. And then what we do is stimulate input A, wait 10 seconds, stimulate B, A, B, A, B, separately, independently. Then we activate them together when we make the postsynaptic cell become activated, what's called depolarization. And we stop, and then we test A, B, A, B, A, B. So just an alternating experiment. That's what all this shows. And when that experiment is done, first thing we noticed was this. So once again, we're showing the size of the synaptic response here. This is during the conditioning. The gray and the white represent the size of the A and the B input, and this is after. And look what happened. For pathway A onto the same cell, the synaptic response got smaller, and for pathway B, it got smaller. Here they are. They did the same thing. So if we stop there, and those were all the data, there's the conclusion. The neuron controls all the synapses do the same thing. However, that wasn't the case as we went through more and more cells. This is an example of a single cell where one input depressed and the other input potentiated onto the same cell in response to the same conditioning. So we did this over and over on multiple nerve cells, multiple neurons, and this summarizes it. And basically what this is showing, if you look at the top here, it's showing the different types of outcomes. The black is potentiation, LTP, the gray is depression or LTD, and the open bars show no effect or no change. Long story short, what we found was the outcomes were no different than if you randomly assigned and tested any one pathway. In other words, there was no evidence to suggest on a single cell the plasticity was yoked together. Each set of synapses could behave the way it wanted, and based on its history or something else about it we didn't know. The only thing else I want to add here, I hadn't really mentioned this, but as Anthony said in the introduction, we do our work primarily in the visual processing part of the brain, what's called the visual cortex. So as part of this same experiment, we also had animals that did not have visual experience. And so these were animals that simply didn't see anything for quite a while. And when we did the exact same experiment with them, all of a sudden, we had all of these plasticity responses yoked together, as shown here. And what that told us is, when the animal is born, when the brain starts, there's some kind of a yoking mechanism in each neuron that tells most of its synapses how to behave when it comes to plasticity. With age, development, and in this case, visual experience, the synapses break away and they become independent computational units, adding much more complexity to the overall computational capacity of this circuit in the brain. So in other words, these outcomes for plasticity can vary between similar cells, and visual experience tends to yoke them, uncouple them once they've grown up. So we need to understand better the mechanisms there. Not sure what happened there. Okay, so now I'm getting to the third part about calcium. So this is work that has been done by two folks in my lab, Quentin Nusser and Haja Kalakulov, worked with me for many years. Obviously, we all know there's lots of calcium in milk. We all know calcium's a fascinating, I mean, it is a fascinating atom. Come on, it's about the coolest atom you can imagine, right? So calcium does so many things in biology. It's just ubiquitous in function. And it can come from a lot of places. We can get our calcium from lots of different sources. You go to the doctor and you talk about your diet, or go to a nutritionist and they tell you, drink milk, eat eggs, do all these wonderful things to get calcium. And that's great. And your body tightly regulates calcium. The amount of calcium that's floating around in your blood, it's not like you're going to change it dramatically depending on vitamin D levels, and there's some pathology. But the point being, calcium comes into our body from a lot of different places. Well, as it turns out, calcium comes in to neurons from a lot of different places as well. And I don't mean milk, cheese, and eggs. I mean different pathways into the neuron that are summarized in this cartoon. So the little yellow box here is a neuron, a nerve cell. The things all around it here are the different pathways for calcium. So there are different types of what are called receptors. You heard about those already. These are what are called NMDA receptors and AMPA receptors. Some of those let calcium come in the neuron. There are also these channels through the membrane, what are called calcium channels. These are what are called voltage-gated calcium channels. When the nerve cell becomes electrically unstable or depolarized, the channel opens and calcium will rush in. Your heart is full of those types of channels, but so is your brain and other tissues in the body as well. And then there's other types of receptors on the neuron that are called metabotropic receptors. And the way they work is a little different than these receptors that are simply channels that open up and let calcium come in. They have signaling pathways where they talk to other molecules in the cell. They release a signaling molecule, in this case it's something called IP3, that binds on what we call intracellular stores here inside the cell, and that releases calcium. And yet there's other stores called ryanodine receptors that also occur in muscle cells that release calcium. So in summary, a neuron has a membrane, and that membrane has channels in it that can let calcium come in. And they can all be pharmacologically interfered with by rather selective pharmacological inhibitors. There are probably a good percentage of people in this room right now that are taking at least one of these calcium channel blockers for high blood pressure, for a heart arrhythmia, or whatever it might be. They're very commonly used in the cardiovascular space, not as much in the brain space, but they have these effects. And then that calcium can be released inside the cell. So what we wanted to know was these types of plasticity that we've been studying, what role does calcium from different sources play? And is calcium really just calcium, or is there something special about it? And so the way we do these experiments, slightly different than before, it's what we call a spike timing plasticity experiment. So this is the neuron, this is the little pipette that's recording its electrical activity. We're stimulating synaptic inputs to the neuron. We record a synaptic response, just like I've showed you before. And then what we do is we activate the synaptic input at the same time that we depolarize the postsynaptic neuron and make it fire action potential, shown here. That's called a pairing. And we stop, and then we analyze the synaptic response before and after. So it's a slightly different protocol for kind of in the weeds reasons. So if we do that in this experiment, just do a control, this is on whatever it says, 30 cells or so. It shows the means and the variances. You can see the baseline average response, the response during the pairing here, and then the response after. So for this group of cells, they actually went up. Their synaptic responses potentiated about whatever that is, about 9% LTP. Not very much, but a lot of variability. You look at the standard error bars here. But this is the most interesting thing. If you look at the individual neurons, they don't look like that average plot. They're very different. This specific neuron, I get 2 green lasers on there. Doesn't get it. Wow, okay. Okay, this specific neuron didn't change at all. This one potentiated. This one depressed to the same exact pairing. So there's something different about those cells, some state they're in, where the same amount of activity, the same kind of activity, the same timing makes one go this way, one go that way, one not do anything at all. And these data are summarized here. So what we're plotting here is the change in the ratio of the size of the synaptic response after versus before, post to pre, the number of observations. The black bars represent those that didn't change, the red ones that are statistically significantly potentiated, LTP, and the blue ones, those that are significantly depressed, LTD. So you can see the distribution in the control animals. So what happens when we start to muck around with the calcium? In this case, we're using compounds that interfere with what's called the metabotropic glutamate receptor, something called MPAP. In this case, we're using a toxin from a sponge we put inside the cell that interferes with the second messenger system to the intracellular store release of calcium. In either of those cases, the group data show potentiation. And if you look at the individual cell data, except for one case right here, it's all either potentiation or no effect. So this is taking this one source of calcium out, the metabotropic receptor binding glutamate, signaling through IP3 inside the cell to release calcium. You take that part of the calcium out, leave everything else intact, and you've completely shifted the plasticity distribution response of the cells. And then if we look at some of the other calcium sources, so this is something that blocks what are the NMDA receptors. This is a drug that blocks the voltage-gated calcium channels. And this is something that blocks what are called the ryanodine receptors in the cell. In each one of these cases, you get a complete net depression, no potentiation. Look over here at the individual cells, no cases of red, everything is black or blue. So taking out the calcium coming through the membrane, through an NMDA receptor, through a voltage-gated calcium channel, and when it hits the ryanodine receptor in the cell to release calcium, that whole pathway is involved with potentiation, which is now gone, and everything goes the other way in terms, in terms of depression. And then finally, what I'm showing here is some combinations, basically, and what we did was block the metabotropic receptor pathway and the NMDA receptors, and here we blocked the ryanodine receptors and the IP3 receptors in the cell. And as you can see, what happens, that pretty much squeezes the plasticity from both ends. There's virtually no plasticity left, a little bit, but not much. So that tells you a couple things. Number one, we of course validated the important role of calcium for plasticity in these cells. Number two, there seem to be two major streams using calcium in different ways that somehow access machinery in the cell to trigger these different types of synaptic plasticity responses. And these data are summarized here. I know it's very busy, but I'll just show you here. This is the control data here. You saw at the very beginning where we got some potentiation. And then here we're blocking with MPEP or zestespungin. Everything goes up, it's potentiated. Here we're blocking NMDA receptors, voltage-gated calcium channels, ryanodine receptors. Everything goes down. And here we block both pathways, it basically levels off. So you have these competing, if you will, calcium influxes and releases. And the real question that comes up is, how do they interact with each other? And what happens downstream? Remember, the downstream plasticity I told you about earlier can be one of two things. It can be putting more receptors in the postsynaptic neuron to capture the neurotransmitter. And or it can be more effective release of neurotransmitter from the presynaptic neuron to bind to the receptors to make a big synaptic response, or for depression to make a less synaptic response, less strong response. And so exactly how these interact, we don't know. We do know that cells are not just bags with everything mixed around. Things are very compartmentalized. They're in very special places in the cell, and these ryanodine receptors and these IP3 receptors sit in proximity at the synapses near some of these channels I told you about. And so we think that part of this is due to the actual physical location of the calcium sources next to the plasticity induction mechanisms. So in summary, there's quite a lot here. So the induction protocol gives variable plasticity outcomes for individual cells. We don't get potentiation when we block either the NMDA receptors, the voltage-gated calcium channels, or the ryanodine receptors. We don't get depression when we block the metabotropic pathway, including the IP3 receptors. And then when we simultaneously block these pathways, we eliminate the plasticity. So these multiple sources interact in the postsynaptic neuron to activate some kind of downstream signaling mechanism. And it still remains to be determined why these calcium signals are differentially dominant in one cell versus another. Why did one start showing potentiation or the other depression? Was it some experience it had? Were those synapses already in a state where they were potentiated or depressed? That very well could be. We don't know the history of these cells or synapses coming to the experiment, so we need to be able to control that, and that's left for future experiments. So basically, there's a lot of things left to do. For the future, one of the important things, I think, is to try to identify optimal stimulation patterns for treating brain disorders. I very much want to try to encourage my clinical colleagues to take these into account. We actually have collaborated with one neurosurgeon out on the West Coast who's trying some of this right now with patients. We'll see if it makes a difference. Hopefully we can figure those things out. We still have the question how individual synapses communicate over space, widely distributed with synapses out here on this dendrite and here. How are they talking to each other about their plasticity state, particularly in the newborn animals until they dissociate? And we'd like to know if we can modulate these calcium signals to optimize brain performance and prevent disease, obviously. So the translational aspects of this are obvious. We need to move the basic science even further to understand the process. And then finally, can we learn from our cohabitants on the planet processes that might help protect our brains from the impact of climate change? I didn't say much about it. I showed you a quick little slide way back at the beginning with the Antarctic fishes. As it turns out with these fishes, they're called ice fishes. They don't have hemoglobin in their blood. They're the only vertebrate that doesn't. They live in virtually freezing water, salt water that's minus 1.8 degrees centigrade. And that water is heated up to about 2 degrees or 1 degree centigrade, which is still extremely cold to your touch. What we've shown is their brain is suffering heat damage at those very subtle temperature changes. And one of the things we know from the early work that we published is what animals do in response to changing temperature around their brain. They lay down different lipids or fat molecules in the brain, some that are more or less fluid. So when they hit a temperature extreme, they lay down more fluid fatty acids in their membrane that give them more mobility. In the more middle temperature ranges, they're stiffer. The membranes are actually stiffer and less fluid. And so animals on Earth have learned to adapt to this. We like to think because we're homeothermic, i.e. we regulate our body temperature internally, we don't have to worry about that. But just look at some of the data recently from emergency rooms of patients showing up in Phoenix, Arizona with what their core body temperatures are, the kind of temperatures people's brains are experiencing, and we need to think about ways that we can try to come up with appropriate interventions going forward. So with that, I'd like to, what do I do here, Brad? Click again. Okay. So there is a symphony. And brains can not only write symphonies and make symphonies, they're playing symphonies inside all the time. Granted, it doesn't have that kind of pitch in those instruments. It's a bunch of clicks and action potentials, but it's a symphony, and it's a coordinated symphony. And the plasticity that happens at all those different sites are the source of our creativity, all the good stuff we do, the bad stuff we do. They even make Donald Trump and Kamala Harris different. So whatever it is, brain plasticity is a symphony for better or worse. And thank you for your attention. I'd be glad to answer
questions. We have some time for questions. Yeah. Questions?
So I had a question about your spike timing plasticity protocol. When you did the pairing, was it always at regular frequencies that you were pairing them, or did you try the irregular frequencies as well?
So that, that particular experiment didn't do anything with frequency per se, it's timing. And what I mean is, it's the timing between the presynaptic impulse and the postsynaptic action potential we generated in the cell. And we would set that timing to be the same delay. It was actually 7 milliseconds. That's neither here nor there. But in other words, we had a very brief time between the presynaptic input and the postsynaptic output that we generated. And it's been known for years that this particular protocol called spike timing dependent plasticity can cause one type of plasticity if you have pre versus pre-preceding post, versus another with post preceding pre. What has not been known is even when the time is the same, the individual cells can behave differently. And that was the point I tried to make. When you look at the group data, which is the way you see it in most of the publications, it always shows, some net effect like potentiation or depression. It's only when you tease the data apart and look at each individual cell that you realize they behave differently. And it's not a hard and fast rule that when the timing is this, it'll always be potentiation or depression. So there wasn't a periodic activation? No, the pairing was just done once every 10 seconds over a period of a minute. Boop, 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 like that. And that, just to add to that, that really comes from psychology, this whole design, the idea of what's meaningful information to the brain when we learn things. When the input is strong enough to drive the next cell in the chain, it must be important. That's the idea behind that. And so you want to strengthen those synapses. When they're not correlated and the postsynaptic cell is kind of firing on its own, that must be noise, and you want to weaken those synapses. That's a very simplistic view, but that's where that theory came from initially. It's called Hebbian postulate for plasticity.
Great talk. So my question is about potentiation depression. As we getting old, we always lose the ability to remember and learn. And so is the learning process always associated with a potentiation, so you have a better memory, better learning capacity, or it's the magnitude of the potentiation depression?
So there are people that study long-term potentiation for a living. They publish lots and lots of papers about it. And some of them would say to you that LTP, long-term potentiation, is really the substrate of learning. And they would say to you that long-term depression is a substrate for forgetting, if you will, or clearing the registers. I think we've all in this field come to realize that's more than a little too simplistic. No doubt about it. I cited some of the clinical cases, but there's lots of really good experimental data in the lab to show if you interfere with this process like long-term potentiation, you interfere with the animal's ability to learn. It clearly plays a role. Depends on the type of learning and which part of the brain and so forth and so on. But there's a lot more to it. You're strengthening the synapse. It's responding more strongly, but how is it discriminating that input from other inputs? And once you think about the whole circuit, so it's a step. It's an important step in the process, just knowing that these synapses are variable. It was thought a long time ago that they were just, every time the synapse went, it went the same. So again, a classic textbook study for medical students is you teach medical students about the neuromuscular junction from the alpha motor neuron in the spinal cord to the skeletal muscle cell. And every time that alpha motor neuron fires an impulse, the skeletal muscle cell depolarizes and contracts. That's the way you want it. You don't want your neuromuscular junction making decisions for you. Once your brain has said, move, get out of the way of the train, you want to get out of the way of the train, right? But in the central nervous system, in the brain, where all that decision making and computation is going on, you want to have variable up and down regulation of synapses, and the LTP, LDD phenomena clearly contribute to it, but I personally don't think it's the whole story. Carla.
Your experiment with animals and changes in temperature. So is there any experiment that shows that there is an effect on learning or behavior as result of changes in temperature?
Oh, yeah.
But mammals, they have temperature compensation. So
Yeah.
how is that the temperature compensation and the effect on temperature in behavior relates?
Yeah. So those papers that I cited up there, that's exactly what we did. We acclimated animals to different temperatures to begin with, 5 degrees C, 15 degrees C, 25 degrees C. We let them stay at that temperature for several weeks. Then we ran all those animals in all the experiments, the biochemistry experiments looking at membrane fluidity of synapses, the electrophysiology experiment recording from the neurons, and behavioral experiments as well. And we showed the expected adaptation in those curves. So the animal, when it's acclimated to a different temperature, you shift the behavioral effects with that temperature. You shift it up, obviously, in the case of the 25-degree animal. So the kind of behaviors we looked at were not very sophisticated behaviors, just swimming coordination and things like that. But we could see it. It was pretty obvious. Now, with the Antarctic fishes, Ishkander and Jordan did a much more sophisticated experiment. They mounted all these infrared cameras around the tanks for the ice fishes, and we got very quantitative data on every aspect of movement of their fins and breathing and all these kinds of things. And we would see them go into the heat effects at 2 degrees centigrade in terms of their behavior. And if we went higher, they were very likely to not be able to eat or reproduce. I mean, there's big concern, as an aside, these fishes represent 90% of the biomass in the Great Southern Ocean around Antarctica, and they're dying off because it's getting too warm. They can't reproduce, they can't eat, et cetera, et cetera. Their behavior is being affected.
I was really fascinated by what you showed at the end with the blocking different types of calcium release, having a differential effect on potentiation. And then the presumption that that's spatially driven. So what I wonder then is, do you see differences in response based on the gross anatomy of the neuron, like with unipolar versus bipolar or multipolar neurons and those sorts of responses?
Yeah, it's a great question, and I'll just give you the flat-out first answer. We don't know because we restricted our experiments to one type of neuron to make sure we didn't add another variable. So every one of those experimental results I showed you, representing years of work, are from what's called a layer 2-3 pyramidal neuron, same morphology, same part of the brain, et cetera, et cetera. So fair question, don't know the answer.
Thank you. I'm approaching this from a public education point of view, high school. I'm personally fascinated by all this, so that's why I attend these lectures. But again, simplest question here. You put up a graph that talked about implanting electrodes into the brain to measure the level of synaptic elasticity. I think I got that right. It was a bunch of graphs, but then you also played an audible version of that.
Yeah, the audio that you heard, that was
Yes.
the stimulation.
So my question is, when, I guess as science goes, there is a graph that's generated, like a visual thing, and then there's the auditory way of thinking about that. Are those two separate processes in science? And also, is there like some, I don't know, for folks that might be blind, is there a tactile version of that kind of thing? This is more like, how can other folks interpret the information you're offering?
So you raise a really fascinating question. It gets into a different subject, but I can comment on it. Briefly. As it turns out, the reason that people like me who have been doing this for years use auditory signals to listen to these things, and even if you talk to neurosurgeons, what you'll find is they're listening very carefully on a loudspeaker in the operating theater, is our ears are so incredibly sensitive to very small differences in timing of sound. You can pick up on that little, like, you know what I mean? Just when you're looking at those things, strings of those recorded on the computer, it's really hard to see if there's a little jitter. So it turns out that just happens to be something our brains are very good at, and we as scientists make use of that tool to do the experiments. It's that simple. Well, you can, yeah, the computer analyzes it for you. So we track every one of those impulses, and the computer measures it to within a millionth of a second. So you can just get a printout nowadays with the computer. But what we call online, when you're doing the experiment, before the computer gives you all the analysis, hearing it gives you some immediate feedback as to what's going on. So.
I have a question.
Okay.
This is a sort of top-down question. You know, there's been this constant argument that either the neuron individually knows and then lets its neighbors know, or that information really doesn't depend on a single neuron. It's the network. And so, you know, based on the sum of your experience thinking about exactly this problem at a mechanistic level, does the neuron know anything, or does it only know to tell its neighbors something so that the network can know something?
Yeah. So what Anthony is referring to is really an age-old question in neuroscience. Nobel laureate Torsten Wiesel won the Nobel Prize about 40 years ago or so now, who studied neurons in the visual cortex, was once asked whether there was something called a grandmother cell in the brain, and whether there was an individual neuron on your brain when your grandmother appeared would fire a bunch of impulses and go, that's my grandmother. And he kind of chuckled at that and said, you know, I don't think so, not likely. As it turns out, fast forward, we now know there's an area of the brain that they weren't studying that is involved with what we call facial recognition. And there are neurons that more or less can figure out different faces. So even though it seemed like crazy at the time, maybe not as crazy, but I think I would say to Anthony's question that I would strongly believe, for a lot of reasons, that really it's the overall network activity. And the synapses are part of that network, and that's changing. In any moment in time, like the picture of the symphony, which synapses are part of this network is changing based on their dynamic up and down regulation. So your brain can be in all these different states with the same physical hardware. It's got it all over our computers in that respect. So
if there are no more questions, I really want to thank Mike for a really superb talk.
thank you. Thanks, Nathan.